In this experiment, you will be shown the stages of protein biosynthesis in the cells of eukaryotic organisms. A cell is the basic structural and functional unit of any living organism. In eukaryotic cells, there is a double membrane organelle called a nucleus. The nucleus contains hereditary material called the genome. The human genome consists of more than 20,000 genes. Each chromosome consists of long chains of DNA molecules wound on specific histone proteins. The DNA is made up of specific sections called genes. Genes are instructions for the formation of a variety of proteins. One gene codes for one protein. The process of protein formation is called biosynthesis. The enzyme RNA polymerase detects a promoter region on the DNA strand. It attaches itself to the DNA in this area and begins to move along the strand. At the same time, the double helix of the DNA molecule unwinds, and RNA polymerase reads information from only one DNA strand. Messenger RNA, or mRNA, is synthesized by the nucleotides that get into the DNA matrix from the nucleus. The hereditary information of the DNA encodes the nucleotide sequence. The synthesized RNA completely replicates the structure of the DNA. This process is called transcription. The transcription process expires when RNA polymerase reaches the terminator section. Before an iRNA can be used as a template for protein synthesis, it must be processed. This process includes the removal of introns and the joining of exons in the synthesized molecule. Messenger RNA leaves the nucleus and enters the cytoplasm. The gathering of amino acid molecules is called translation. This stage of biosynthesis takes place in non-membranous organelles of the ribosome. The initiation step involves the attachment of the ribosome to the messenger RNA strand and recognition of the start codon for protein synthesis. Transfer, or tRNA, transports amino acids to the site of protein synthesis. Each transfer RNA has an anticodon that binds to a specific codon of messenger RNA. If they are complementary, it attaches its amino acid to the former strand. Synthesis occurs according to the genetic code, i.e., a specific amino acid corresponds to each triplet of mRNA nucleotides. Elongation occurs, a buildup of the polypeptide chain of the protein molecule. The 20 basic amino acids are involved in the formation of the protein. The last step is termination. Protein synthesis ends at the so-called stop codon. Finally, the amino acid chain is compacted into a protein molecule. So, protein biosynthesis takes place in several steps. The first step is transcription. The second step is translation, consisting of initiation, elongation, and termination.